We're told in Scripture that all created things have a glory. That is, all created things give glory to God in their own unique way. Today I'm going to be talking about giving you snapshots of what's often called the new biology, which includes Evo-Devo, the extended evolutionary synthesis, but also bioelectricity and other developments. I have a limited amount of time, but I hope to give at least some structured impressions of the purposefulness of life and how it challenges more deterministic interpretations of biology. Interpretations of biology inform our sense of natural law and bioethics, even for secular folks where it might happen unwittingly. For us Catholics, I would argue it's even more critical we get things right about biology. I see this as a possibility to re-enchant a Catholic cosmology which sees living things to be the intricately wondrous things they reveal themselves to be. A proper account of life gives the due glory to God and brings us toward Him in this contemplation and stewardship of creation, and I feel like this connects well with the homily this morning, which I thought was very good. But let's start at the beginning. And so in the beginning was the Logos, in the preface to the Gospel according to St. John. The beloved disciple reveals what was veiled in the first creation account of Genesis. In the beginning was the Logos, the Word, that is the reason and intelligible order underlying the cosmos. The Logos is the author of creation, and He was made flesh and became man. We humans, being made in the image of God and having the faculty of reason, can perceive and know aspects of this reasonable natural order. Namely, we can know and understand science. And this is what Ross Douthit was even talking about on Friday evening. And irrespective of philosophical positions that secular scientists may have, they have to accept, at least on some kind of faith, that the world is intelligible, and this knowledge is somehow accessible to our intellect. Because if it weren't for this, we simply wouldn't be able to figure anything out. And so in my experience, when pushed on this question, scientists often simply accept this as a brute fact. Catholic scientists, on the other hand, can provide a reason for this, namely the divine reason, or the Logos. And so let this be an image of faith and science. There's a minimal view which we can discuss and debate on its own terms, but the higher order Catholic view provides the more complete account. They're more like concentric circles rather than Venn diagrams. And so in the first creation account of Genesis, all the different creatures are described as being brought forth by the earth and the waters, each after its kind. Kenneth Kemp also was discussing this in his talk. Some of the church fathers, like Augustine, argued that God created the world instantaneously and had embedded His plans for creations in the very fabric of the universe. Every plant, all kinds of animals, swimming, creeping, or flying, were forethought in the mind of God, and these plans were infused into creation only to be brought forth according to their kind in due time. And so in his book on the literal interpretation of Genesis, St. Augustine developed this idea, building on the Stoic concept of rationes seminales, which literally means seeds of reason. And so the analogy was made with a seed and how an acorn, in this case, contains the potentiality of the oak, but only germinates and becomes the oak when the conditions are right. And so similarly, God created everything in seed form at the beginning of creation, embedded within primordial matter. And over time, these rationes seminales were actualized or brought forth by secondary causes within creation. And so although the church fathers were not cryptically talking about evolution, some of the earliest Catholic responses to Darwinism in the second half of the 19th century brought up how the rationes seminales could help explain how new species emerge after the 6 days of creation. St. Maximus the Confessor developed a parallel concept from the Neoplatonists with the Logoi, which is the plural of logos. Although God doesn't have parts, the Logoi are like little pieces of the Logos insofar as they exist in the mind of God. The Logoi are essentially the rationes seminales, but with St. Maximus, there's less emphasis on the creation account, let alone creation of new species. Instead, he focuses on the centrality of the Creator Logos and how His will is to draw all creation to Him. For us humans, this, of course, is theosis. But St. Maximus emphasizes how all the Logoi, not just humans, are drawn towards Him. In St. Maximus, we have a more mystical account that emphasizes how all creation groans for everything to be put in its right place. And in evolutionary history, we see many forms being brought forth. Many of you will be familiar with convergent evolution, which is the observation that several independent lineages have produced several similar features, such as body forms, body parts, and lifestyles. A book on this subject cleverly twists Darwin's famous line about endless forms most beautiful to limited forms most beautiful, because you see certain forms are prioritized or And the main idea how this arises, though, is that there are limited optimal solutions for adapting to ecological niches. Whether it's the streamlined bodies of dolphins or ichthyosaurs or also sharks, or it's flight in birds, pterosaurs and bats, or the camera eye in several lineages, including humans and octopuses. The properties of water, air, and light strongly constrain and limit the possible adaptations for specific lifestyles, and thus evolution progresses not as haphazard and directionless as often is spoken in evolution. And Simon Conway Morris has written extensively about this, including the directionality of evolution, and he famously rejected Stephen Jay Gould's account of evolutionary contingency, which stated that if you rewound the tape of life, you would get something completely different, implying that humans would not even come out of that process. And Conway Morris suggested that with some differences, the same forms would be brought forth. It may be easy to see how natural selection could select the most streamlined body, the most aerodynamic wings, and the best camera eyes to see. But how do these things come about in the first place? And so it's often been said that natural selection can explain the survival of the fittest, but it cannot explain the arrival of the fittest. Natural selection itself doesn't do anything, it just selects the winners. And so the neo-Darwinist account essentially posits that small random mutations over large stretches of time can account for the arrival of the fittest. Sometimes it's a little more sophisticated, but often it's not. And so I should point out that many, if not most, of the critiques of evolution from the intelligent design community are critiques of neo-Darwinism. For example, they point out the problem of waiting times, namely, given what we know about mutation rates, there hasn't been enough time to account for the genetic diversity that we see. And they're not wrong in many of their critiques of neo-Darwinism, or what we might call the old biology if the other one is the new biology. And so the old biology remains to be the dominant interpretation of biology, however. It's centered around the gene as a fundamental unit which can explain everything in a living system. Think here of Richard Dawkins' selfish genes, but also like when E.O. Wilson said that the organism is only DNA's way of making more DNA. And this is what C.S. Lewis called nothing buttery, which is a tendency of many scientists. Oh, X is nothing but Y. And so likewise, an organism is nothing but genes. Descartes is famous for reducing animals to nothing but automata, and here we have one of these automata from those days. And I have here representing the mechanistic view of life that was renewed with a gene-centered view of life. And so the fable of science recounts that modern science did away with formal and final causes, in no small part due to people like Francis Bacon and René Descartes. And of course, the Thomist bugbear of nominalism is responsible, but we won't get into that here. And so formal and final causes are among the 4 Aristotelian causes. Formal causes are not just what we call the form of a thing, like the shape of a thing. It's rather what makes a thing what it is. But I'll return to this in a little bit. And final causes, the telos in my title, are more often known under the name of teleology, which, roughly speaking, refers to how things are directed towards ends or goals. And even though these were supposed to have been tossed out, biologists are tempted by teleology. J.B.S. Haldane is famously quoted as saying that teleology is like a mistress to a biologist. He cannot live without her, but he's unwilling to be seen with her in public. But it's inevitable. Goals, purposes abound in the language of most molecular biologists. Nitrogenase is for fixing nitrogen, rubisco is for fixing carbon, but these goals are much larger in scope. The goals of these enzymes belong to the organisms that express these enzymes and use them. When we look at a simple single-celled organism like an amoeba and how it can hunt prey, respond to lights, sounds, and smells, and move in sophisticated ways without the benefit of a brain, we realize that the different genes and their products are being used towards unified goals. And so little by little, scientists who are not particularly religious have begun to rediscover metaphysics in partial ways. It might have started with cybernetics and systems theory, which led to complexity theory with concepts like emergence and downward causation, which are very teleological concepts. To avoid the usual accusations of Mysterianism hailed at anyone who brings up teleology, they'll change the language sometimes. Instead of teleology, there are discussions of teleonomy instead. But it's still there. And so it's not just teleology that's really gone away. This line is from Richard Lewontin from an essay in the 80s critiquing the gene-centric view. It didn't really catch in those days, but there's an increasing return to the organism as a unified thing that exerts downward causation on its parts, which I hope to convey. And this is the way in which formal causality hasn't gone away. life has a very special substantial form that is life, which Aristotle called the soul, which is completely different from the formal cause of non-living things. In this framework, living things have immanent causation, which David Oderberg defines as something, causation that begins with, and I'm saying with the organism, terminates in the organism for the sake of the organism. And these are three of Aristotle's causes, of course. And so non-living things, in contrast, have transient causation, and this distinction is captured by William James, who compares how Romeo wants to be with Juliet, kind of like how iron filings want to be with the magnet. And if there's nothing in the way, Romeo is going to go straight to Juliet, just as the filings will go straight to the magnet. But if there's a wall between them, Romeo isn't going to press his face to the wall like an idiot. He's going to find another way, like scaling the wall to kiss Juliet. And so, notwithstanding the questions about the origins of life and the origins of cells, which I'm not going to get into, but once there were cells, single cells, there must have been the ability to problem solve at various levels, as we see in the simplest single-celled organisms. And there's an adaptive value to this, and this is related to the idea of the Baldwin effect, that the ability to learn new behaviors has tremendous adaptive value. The single cell is the simplest form of life, which is why I start here, because its competencies scale up to the multicellular organisms. And Barbara McClintock, who's pictured here, she won the Nobel Prize for her discovery of transposons. These are the so-called selfish genetic elements that are the jumping genes that you might have heard of. They make up about half of our genome, and they're sometimes called junk DNA, and they have all these other interpretations. May's actually has about like 85% of the genome is transposable elements. And in her Nobel lecture, she said these things. She was talking up here about the ability of a cell to detect chromosomal damage and repair it. And so she said, cells make wise decisions and act upon them. And then later in the talk, she said, a goal for the future would be to determine the extent of knowledge that the cell has of itself and how it utilizes this knowledge in a thoughtful manner when challenged. And she's not talking about the cell's conscious or anything like that, but she's talking about that there's a lot more that's happening with a cell that is, less determined by the genes. And so we start with a little refresher of biology, just to get our things straight. And so genes are organized in a genome architecture, and so these are the familiar chromosomes that you're probably aware of seeing. This is the human karyotype, which is just all of the chromosomes lined up together. And so there are pretty big differences between the karyotypes of different organisms. This is actually why the liger cannot reproduce after, is that they actually have, the lions and tigers actually have the same amount of chromosomes, and so it's not a matter of just the numbers game and them pairing up, but it's just that their structure is completely different. And so genes are like bricks in an arch, and the form of the arch is the 3D structure of the genome. In the nucleus of a cell, DNA is tightly packed into the familiar chromosomes we can see here in the karyotype and also here in this other figure. And so these are the beads on a string. And so there are different ways that an organism can regulate the structure of the chromosome, and they can either activate or silence certain genes based on the structure. One is just directly methylating a gene, which silences it. The other way to silence a gene is actually to tightly bind it in heterochromatin, and so this DNA is so tightly bound that the transcription apparatus cannot get inside to transcribe any genes. And then we have euchromatin when it opens it up in order to transcribe it or to do some other process. And so these are the sorts of modifications that are needed. And so genes actually need some sort of other drive to try to turn on or silence genes. And so in order to transcribe a gene, you have to be able to recruit the RNA polymerase in order to transcribe and make RNA. And so how this is done is there are transcription factors, which are proteins that binds to certain regions that are generally upstream of the gene that it's going to promote the transcription of. And these transcription factors can bind to different regions upstream, and they can either activate a gene or they can repress. This is the LAC-Z operon, the LAC operon. And the elements that they're binding to can be of all sorts of forms. It's not just the promoter region. There are enhancers, there are silencers, there are insulators, and all these can regulate either positively or negatively. And I actually forgot to mention, these are the symbols in a lot of these gene circuit diagrams that geneticists like to use. This is to activate, and this is to repress. And so you can already see that you have this system where you can have complex gene regulatory networks that something might activate something, and that might have a negative feedback to repress the thing that activated it, and so on and so forth. And when you scale this up, you actually get these gene regulatory networks that drive all of development, and they drive actually so many other processes. They're able to organize these different genes into submodules that can be then edited in fine-tuned in different ways. And so these are the famous Hox genes in Drosophila. This is kind of like one of the typical cases that we find in Evo-Devo. And so there are different modules that are responsible for different parts of the fly in this case. And so one of these, just looking at one of these, you can see that there are all sorts of downstream targets of this one Hox gene that it's turning on different things in order to develop the proper morphology at that spot. And so you can have all sorts of different gene regulatory networks driving different things. And this is actually how diverse body forms largely are generated using developmental affordances. This is the whole Evo-Devo. So many, humans and nematodes are formed in just modifying some of these different gene regulatory networks. And so alternate splicing is this, it shows how transcription is modular, how you can form different proteins, protein isoforms from different exon combinations. And so it's very, transcription is very modular. And organisms aren't really passive victims of that selfish DNA that I was talking about. They can actually turn off transposons using both DNA methylation and histone packing. And they can actually repair certain areas more often than others. There are certain histone marks that recruit more DNA repair enzymes to specific loci. And so organisms have domesticated transposons. They can jump and become promoter regions, enhancer regions, silencer regions. They can help, sometimes because transcription transposons are methylated, they can silence a gene. They can be an exon. They can actually be a retrogene. They can actually sort of insert an already spliced mRNA, and they can insert it into a gene. And so you can have this modularity that is far more complex than just single point mutations. In cancer, you find that some of this remodeling it forms what this Henry Heng calls genome chaos. And so you see the normal karyotype here, and so each chromosome is colored with a specific color, but then you see that it's completely reorganized and it does all these rearrangements. And so Henry Heng has actually proposed this as a model for understanding how macroevolution could take place and how you could explain the punctuated equilibria that you see in the fossil record, that you see all of a sudden these like quick jumps into something completely different, and again that doesn't observe the normal waiting times that we would imagine were the case with just the single point mutations. And so the idea is that you can have this drastic chromosome reorganization that is macroevolution, and this is how you can get different karyotypes that are then incompatible. And so oftentimes, some people like Henry Heng say that the karyotype is kind of a definition of the species. And so then after, there's this macroevolution, then you can have the microevolution. And I'm not denying all that other stuff is happening, but then you can kind of explain how things kind of get back together, because this is obviously not some sort of entirely purposeful thing. And all these things happen when cells are under stress and when they would be in more need of adaptation. And so here's some books that talk about that. Cells, when they're perturbed, these are salamander cells in the kidney. They form these tubules normally. When they're haploid, when they just have one copy of the chromosomes, they form these tubules. But when you perturb them and make them either diploid or polyploid, in this case pentaploid, they form the same size of tubules. And so even though the diploid is larger than the haploid cell, they still manage to arrive at the same size of tubule using less cells. In fact, when you get a pentaploid, it just uses one cell and it wraps around itself. And so it's using other mechanisms within the cell to achieve the same goal. And so it's arriving at something different. It's like Romeo finding some other way to reach Juliet. And here's an experiment that Michael Levin has done where they completely scramble up the face of a tadpole, and it remodels it as needed in order to form a normal tadpole head. It'll find a way. It knows what the set point's supposed to be, and it'll do it. They grafted an eye on the side, and they've actually done on different parts of a tadpole, and it's kind of sad, but they removed the other eyes. But this eye actually developed an optical nerve that got to the brain, and it could see through that eye. And so the organism is able to somehow exert this downward causation in order to accommodate for all these different things. So it makes you question whether or not genes are just deterministically producing these things. The organism is using the genes to do what it wants to do, to reach its goals. And so bioelectricity coordinates all sorts of things. It originates using these ion transporters, which creates this, a charge separation on the membrane. And so you can develop a voltage across the membrane. And it's used in all sorts of things in cells: proliferation, migration, differentiation. It's used in tissues. It's used obviously in functions like the heart. It's used to organize development into spatially organized development. And it's also used to recruit other cells in wound healing. And so bioelectricity can do all sorts of things. This is also from Michael Levin's lab. Just using bioelectricity, in this case, they micro-inject mRNA of ion channels into different parts of the tadpole. And they can actually generate an eye just based on these bioelectric perturbations. So they'll change the bioelectrical patterns in order to drive the formation of an eye, because that's how the eye is normally formed. And here is just how these voltage-modified cells can actually recruit wild-type cells that are not modified. So they're interacting with wild-type cells. Bioelectrical signals can also this is also from the Levin lab, where they modified the bioelectrical pattern, and they can generate these two-headed planaria. These are like flatworms that generate two heads. They can actually change the bioelectrical signals in a planaria to form the heads of other species that phenomimic the other species. And finally, just the other point on bioelectricity is that cancer cells actually separate themselves from other cells. They cease to cooperate with the organism, and they sever their ties. And so here, the Levin lab, they introduced these strong oncogenes into tadpoles, and it develops a tumor if you just inject these strong oncogenes. Here's the abnormal voltage state that you get as a result. But if you introduce the oncogene with these other hyperpolarizing channels, you can actually stop the formation of the tumor. It doesn't form because the bioelectrical signal is strong enough to exert that downward. And so with that, in medieval science, saving the appearances meant constructing models that fit observed phenomena. The observed phenomena here point to there being more than just genes, and at least cells and multicellular collectives are doing much more of the work than often is recognized. And C.S. Lewis in The Abolition of Man talks about the destructive effect of Baconian science and how this is related to the rejection of the natural law in modern times. He proposed a different direction for science and suggested that perhaps Goethe's ideas on science might be a good start. Goethe emphasized the view of the whole organism in contrast to the prevalent mechanistic view. He had an approach of careful, immersive observation of natural phenomena, which I don't have time to outline. But using this method in studying plants, he predicted that flowers are essentially specialized leaves. And with the advent of molecular biology, the ABC model of flower development has actually shown that indeed all the parts of a flower are modified leaves, which get their identified from specific transcription factors that effectively turn on those modules that determine the sepal, petals, and reproductive parts. And I would say that Goethe's approach is an example of saving the appearances. And so C.S. Lewis also proposes a restoration of natural philosophy, and he doesn't speak to this point as much in depth, but I would argue a return to the sources in Aristotle and St. Thomas. And there's a lot of work in neo-Aristotelianism with a lot of Thomists involved, which is exciting as some of the new biology. But ultimately, when we go beyond the territory of science into metaphysics, we must remember what St. Maximus reminds us of, that as humans, our ultimate end is to be united to the Logos and Theosis, and that we must strive to live virtuous lives towards that end, which for scientists includes how we are stewards to creation in our science and our technological interventions, which also has to do with the homily this morning. And so that's why I think this is important, that we don't just see life as nothing but watches, genes, and survival machines, and instead view the richest of living systems in their entirety. And in this contemplation, we deepen our sense of the glory of the Logos, the glory of God. And I'll just end with this quote from St. Ephrem the Syrian. In his book, Moses described the creation of the natural world so that both nature and Scripture might bear witness to the Creator. Nature through man's use of it, Scripture through his reading of it. These are the witnesses which reach everywhere. They are to be found at all times, present at every hour, confuting the unbeliever who defames the Creator. And with that, I'm done.
In the instance of time, we're going to take one question so we can get back on schedule. Right here.
Thanks for your great talk. I'm a structural biologist, and yeah, I'm interested in your view, a kind of broader vision of causality beyond just sort of mechanistic, this arrow moves this arrow, moves this arrow. What might that look like? Is structural biology necessarily reductionistic, or what might a different, broader view of causality look like in structural biology?
I mean, a lot of what I'm talking about is the way in which things are transcribed and expressed, and all the different regulation that goes on. To like, I guess, in what way are you referring to structural biology?
Like the structure of how an ion channel opens and closes, how the atoms move around.
Right, but I'm talking about what turns those things on, or what sorts of things activate, and not necessarily the structure themselves. And I'm not necessarily saying that like evolution is as intentional and purposeful in the sense that the cell is like specifically designing structures that are going to open, some sort of channel or anything like that. So I'm mostly talking about the way in which genes are expressed, and the different hierarchical forms of causality, like in gene regulatory networks, in bioelectricity, and so on, and in physiology. But that there's this downward causation, that there's something happening, and it could be some sort of mechanistic thing. I'm not saying that it's incompatible with that. But it serves the interest of the organism, and the organism is the ultimate phenotype. Even when you have like other cooperative organisms and stuff like that, like the organism is still some sort of fundamental unit for biology, and that's mostly what I was trying to convey.
Thank you.
Thank you very much.